Hi there. In this video, I'll be talking about DNA mutation, one of the basic parts of evolution. For example, if you didn't have any mutation, there would be no evolution at all. So while many people think that mutation is a bad thing, it actually turns out to be a very good thing. As I pointed out in a previous video, biological evolution is dependent on three main factors. First is mutation, which I'll be talking about in this video. Second is selection, and I'll talk a little bit about that today, and then fill in a different aspect in a different video. And the third is random genetic drift, which I'll cover in a different video. Before I talk about mutations, the nuts and bolts of mutations, I want to introduce a bit about selection and how people normally think about it and how I want you to think about it in this class. Normally we talk about selection as it pertains to the entire organism and its relation with external environmental challenges. However, the selective pressures exist at all levels within the organism. That is, there is selection at the level of transcription, splicing, transport out of the nucleus, translation, etc. within the cell, as well as intercellular selection and selection at the tissue level, all the way up to the whole organism. Of course, there also is selection operating in terms of the interactions of the organisms with its external environment. But that's really the last step in selection, and all of the pressures together determine the overall rates of mutation. So I want to discuss that a little bit here. So I want to talk about the levels of selection and the fixation of mutations. So DNA can be altered, right? You can make chemical changes to DNA, but those are not considered mutations until they are inherited. So a DNA change is just a DNA change. If it has no effect, then it's not a mutation. If it's passed on to its offspring, or its sister cells or daughter cells, then it's considered a mutation. So there's an inheritance filter to fixation of that change in those cells. The next step is, does that mutation become part of an RNA molecule? That is it transcribed? If the mutation affects a promoter or some other region, then it may be fatal and may mean that RNA is never produced. So there's a transcription filter. Next is the RNA spliced and transported out of the nucleus in eukaryotic cells. That is that RNA properly processed? So there's a processing filter. So the next selected filter is a translation filter. That is, okay, it's been exported out of the nucleus. Can it find a ribosome? Is it taken to the ribosome by certain proteins? And is it translated into a protein or a peptide? Next is the protein able to be processed properly? That is it able to become a mature protein and be transported wherever it's supposed to wind up in the cell? So there's a processing filter there. So once it's been expressed all the way to a protein level and it becomes a mature protein, is it still functional? That is, does that protein do what it's supposed to do in an efficient way or in the way that it is supposed to function? So there's a functional filter for that protein. And next, does that protein provide an advantageous function for that organism so that it can reproduce and produce more of its progeny, thus propagating those genes? So all of these initial filters, these selective filters up above, are at the intracellular level within the cell. But there are also way out here, do they provide a function and an advantageous function for the organism or the cell? are happening at the intercellular, extracellular, and extraorganismal level. And so the total mutation rate, going back to the beginning, really includes all these steps, although most people measure whether a mutation is advantageous or not way, way out here at this level. So they're really underestimating, in most cases, the amount of mutation that actually goes on within an organism. Okay, so now I want to focus in on these mutations and causes of mutations. Here's DNA up here. You can have mutagens causing mutations of those DNA, such as UV light, chemicals, etc. Recombination can also introduce mutations into the DNA, as well as microorganisms can change the DNA and cause mutations. And this includes bacteria, fungi, and viruses primarily. Also, some protists can cause DNA changes as well. The second part of this is DNA replication, and even the replication proteins, if they're mutant, can cause mutations in that replication process. Microorganisms can also cause mutations in that process. If for some reason there are some of the nucleotide triphosphates that are in low concentrations, such as guanosine in development of humans, often causes mutations. Also, DNA polymerase slippage and dislocation can cause mutations. And then even if the DNA is mutated, it can be, or changed, it can be repaired. But if you have mutant repair proteins, those repairs will not take place. Microorganisms can affect that. DNA polymerase slippage can also affect repair. And again, low nucleotide triphosphate concentrations can affect DNA repair as well. So here are some of the mutagens that can cause mutations in DNA. First is UV irradiation, a very common one. When you get sunburn, you're actually mutating your DNA. Chemicals, ionizing radiation, infections, and some enzymes. The next causes for DNA mutation are first faulty DNA polymerase or slippage of the DNA polymerase, low DNTP pools, especially guanosine sometimes is low, a primase and RNA primer error, and also a faulty gyrase and helicase that unzips the DNA during replication. And then other proteins such as single-stranded binding proteins. Rec A is one in bacteria and Rec1 in humans. Remember, these stabilize single-stranded DNA during replication. Altered base pairing also is possible. So during replication, you can, A pairs with T and G pairs with C, but occasionally G can pair with T, A can pair with C, and G can even pair with A. Normally, these are caught by the repair enzyme systems, but sometimes they aren't, and so the mutation gets through. And then there are several repair enzyme pathways, and so if those are intact, then often these mutations will be repaired, but there are several different types of repair systems: base excision repair, nucleotide excision repair, mismatch repair, and DNA glycosylases. So if any of these, and they include several different proteins, several different enzymes, if even one of those is mutated, then that can affect the repair. And we'll talk about repair in more detail in another video. So here's the error frequency at various stages of DNA synthesis and repair. So if fidelity of replication was based just on base pairing, then there would be a mutation every 10 to 100 bases. That is, 10 to the minus 1 is 1 over 10, and 10 to the minus 2 is 1 over 100. However, DNA polymerase copies with more fidelity, and it even has some proofreading functions. And so the error rate would go down to 10 to the minus fifth to 10 to the minus sixth, or 1 in 100,000 to 1 in a million. Accessory proteins, single-stranded binding proteins, and others further lower the mutation rate to about 10 to the minus seventh. And then there's post-replication correction mechanisms, repair mechanisms, that bring that error frequency down to 10 to the minus 10th. But it's hard to sort of conceptualize what's actually going on there. So if you look at the number of errors per genome per division in a 10 to the 10th base pair genome, which is close to the human genome is about 3 times 10 to the ninth. So, but 10 to the 10th makes it easier to conceptualize. So the mechanism of base pairing, the number of cumulative errors in that genome would be from 10 to the 8th to 10 to the ninth, or 100 million errors to 1 billion errors in that genome every time the DNA was replicated. So clearly that's not going to work. Even DNA polymerase on its own would make between 10,000 and 100,000 errors every time the cell replicated and divided. So even clearly with that level, our bodies would be full of all sorts of mutations, and we would not live very long past conception. Accessory proteins bring that level down to 1,000 errors per genome, per replication cycle. Not too bad. And then mismatch repair and other repair systems bring that down to fewer than 10 mistakes per that 10 billion base pair genome. So that's quite acceptable. So during evolution, as genomes got larger and larger, from millions of base pairs to billions of base pairs to nearly trillions of base pairs, these systems had to get better and better at detecting and avoiding mutations, even though mutations are one of the basic parts of evolution. So how does this relate to you then? Well, your body contains about 10 trillion cells, 10 to the 13 cells. Each of those was the product of a replication and division cycle. And if, on average, there is one mutation per genome per replication cycle, then you will have approximately 10 to the 13th mutations scattered throughout your body in various cells. The probability of a mutation in a particular gene in a particular cell is about 5 to 10 in a million. So a gene is about 1,000 base pairs, and the human genome, haploid genome, is about 3.2 billion base pairs, and that's how I come up with that number. But the probability of a mutation in a particular gene in any of the 1 trillion cells is much higher. So there's a probability that there's at least one mutation in a particular gene somewhere in your body. So if the mutation occurs early in development, however, it will be inherited by more of the subsequent cells. So that's why, it's very important during pregnancy to avoid mutagens of any kind and have good nutrition so that you have all the nucleotide triphosphates that the developing fetus needs. But what are the most common spontaneous mutations or reactions in DNA? Here are 2 base pairs, an AT pair and a GC pair with the sugar phosphate backbone in each case. So some of the most common reactions of DNA are hydrolytic attack, and the size of the arrow here indicates the frequency of those attacks. Now, hydrolytic attacks can be caused by acidic conditions, such as in your stomach and other places, or if you get acid on your skin or something. So the most common things that happen are that the purines are broken off from their sugar, the deoxyribose there. So then they create apurinic sites, that is, the purines are no longer there. To a smaller extent, the C's and the T's, the pyrimidines, are chopped off from the sugar group on that glycosidic bond. And then there are a couple of other minor reactions, including nicking the backbone of the DNA, breaking the backbone of the DNA. And all of those will, unless they're repaired, will cause mutations if they're inherited after subsequent cell divisions. Another common set of reactions are oxidation reactions. And of course, your cells use oxygen in oxidative phosphorylation. That's why you're breathing in oxygen now. And it's concentrated in the mitochondria. And so these reactions occur more often in the mitochondria. And again, the size of the arrow, these red arrows in this case, indicate the frequency with which those reactions occur. And one of the major reactions occurs with a guanosine, and that's shown over there for the guanosine. And that will cause mutations, and the buildup of mutations causes death eventually of those mitochondria, and death of the cell as well. Another frequent change in DNA, shown here in green, is caused by S-adenosylmethionine transfer of a methyl group. So this is a methionine that can transfer a methyl to the DNA and cause a mutation, cause it to change. This is one of those interesting molecules that is an ancient RNA molecule, S-adenosylmethionine, which normally in cells transfers methyl groups around to different biochemical molecules, especially proteins, but in this case, other nucleic acids. But it causes mutations in DNA. All three types of these DNA changes can lead to mutations of the proteins in the cell, and often they can lead to tumors and cancers as well. Changes or substitutions of the nucleotides occur at different rates. In this figure, the length of the arrow indicates how frequently those changes occur, or those conversions. So transitions occur much more rapidly or readily than transversions. So a purine changing to a different purine, that is an A to a G or a G to an A, change more rapidly than an A changes to a T or a C, and vice versa. You will notice also that the C to T changes happen the most often, whereas T to C changes occur at a rate that's approximately equal to A to G or G to A changes. The main reason for this is that cytosines are often methylated in cells normally to 5-methyl cytosines. And when those are deaminated, they become a thymine. So the frequency is high in a C to T change. The reason that Cs are methylated are for several reasons, actually. First is that they distinguish between a template DNA and a newly synthesized DNA strand during replication. So the enzymes and DNA recognition proteins can recognize the difference between a newly synthesized strand and the template or older strand, and then if they need to make a correction because of a mutation, they will most often change the newly synthesized strand and stay away from changing the template strand. Another reason is that when the DNA is highly methylated, then it can cause a conformational change in the DNA, and it will change it from a B-form DNA to a Z-form DNA, And that normally turns the transcription or the expression of that gene off, and so the gene cannot be expressed. The third reason is to protect the DNA from restriction enzymes, and normally a restriction enzyme will recognize a certain DNA sequence unless it's methylated, and then if it's methylated, that restriction enzyme or DNase will not recognize that site, and the DNA then is not cut. So here's a diagram just to show you how that works. If you have a normal cytosine and it's deaminated, then that becomes a uracil. But the repair systems in DNA recognize that uracil should not be in DNA. So the repair enzymes will chop out the uracil and replace it with a cytosine. However, in the case of 5-methylcytosine, if that's deaminated, it becomes a thymine, which is a normal base in DNA. So it's not recognized by the repair system and chopped out. So during the next replication cycle, one of the strands that's replicated becomes normal again, and the other one then creates a mutation at that site. Another common mutation happens when DNA is exposed to UV irradiation, such as when you go out in the sun, when you sit under sun lamps to get tan, etc. Then you create pyrimidine dimers. And so these are when you have adjacent pyrimidines, primarily thymines will do this. So you have thymines that form a butyl ring together and cause a pucker in the DNA. You can also have TC, that is thymine, cytosine dimers, or cytosine dimers, but they occur much less often. There's a human disease called xeroderma pigmentosum. Actually, it's a series of diseases where there are mutations in the DNA repair system for pyrimidine dimers, and these people go out in the sun and they get lesions and tumors readily, just with mild exposure. They can even be exposed to fluorescent lights and develop skin lesions and cancers. So here's a list or a table of the major mutations of nucleotides in DNA. So if the original nucleotide is a cytosine, then when that's deaminated, that becomes a uracil and it pairs with A rather than G. But there are repair enzymes for that. And then there's the 5-methyl-C. When it's deaminated, that becomes a thymine and it pairs with an adenine. And so that creates a mutation because there are no known repair systems for that. Then an A, when it's deaminated, becomes hypoxanthine, and that will pair with a C rather than a T, but there are repair enzymes for that. G, deamination, becomes xanthine. It pairs with a C, which seems normal, but it's not quite normal, but there are repairs for that. Then the A's and G's can be depurinated, and in that case there's no base, and so it's ambiguous what they pair with, but there are repair systems for that. And then, as I mentioned previously, in the mitochondria, where there's a lot of oxygen around, guanosines can become hydroxylated, and they'll pair then with adenines, and it's not known whether there's repair for that or not. It is known that when you put organisms in high oxygen situations, there are many more of these lesions, so there may not be a repair mechanism for that. Occasionally, adenosines are subject to cyclic oxidation, where you get an 8,5' cyclic A. It's ambiguous how it pairs, and there's no repair known for that. And then dimerization, pyrimidine dimers primarily, no pairing is possible because of the puckering of the DNA, but there are repair systems for that, and I'll talk about that in the next video. And then finally, any of the bases can become hydroxymethylated, and those it's unknown how they pair, and there are no known repair systems for those lesions. And as I said before, much of the studies that I've been talking about were done looking at lesions in cancers, and so a lot of these lesions that I've been talking about occur in many different types of cells, but also lead many times to tumors and cancers. Although some of these certainly could lead to advantageous mutations in particular genes and proteins, leading to evolutionary processes as well. And these have probably been happening since the very beginning of life on Earth. So these mutations can cause different types of effects in those genes and DNA sequences. So I just want to talk about some of the general types of mutations. So if we have this as the original sequence, then there can be a transition, that is a C to T change in this case, which again is one of the most common changes. You could also have an A to a G change or G to A, etc. Those are all transitions. And then an example of a transversion would be this one, which is a G to a C change, which occurs less often than a transition. A G to T change would also represent a transversion. Then there are deletions. So a deletion can be anywhere from one nucleotide up to hundreds of nucleotides. So here an ACCTA has been removed from the middle of this sequence. There also are insertions, so here an inserted piece AAAGC has been inserted to this into this sequence. Often deletions and insertions are the result of a recombination event. Not always. You can get other insertion and deletion events, but commonly recombination results in a deletion and an insertion, a deletion on one chromosome and an insertion on the other chromosome. And the last one here is an inversion. So the DNA has been reversed in one section of this DNA fragment. And notice that the sequence that's been inserted here on that DNA is the reverse complement that came from the other strand. So that's what happens during an inversion. So mutations can happen in any DNA and then be converted by transcription into the RNA version. Now, if the RNA happens to be ribosomal RNA, tRNA, or a small RNA, or an intron, something like that, it doesn't affect a reading frame, but it can affect the structure, the secondary structure of those RNAs. But in mRNAs, you can get several types of mutations that will cause a different reading of the code within that mRNA. So the first one is a synonymous mutation, where the sequence change causes no change in the amino acid. So GTC and GTA both encode valine. So although there's been a mutation, there's no noticeable change in the protein that's expressed from that RNA. When there is a change, it can be a missense change, which can either be cause a minor problem with the protein to fairly major problems in the protein. So the sequence change that causes a change in the amino acid, such as GTC to a TTC, changes the amino acid from a valine into a phenylalanine, and those have two very different functions in a protein. So that may cause a lack of function or a change in function for that expressed protein. So the third type is a nonsense mutation. So in the DNA, you'd have a change from an AAG, for instance, to a TAG that changes the codon from AAG, which would code for lysine, into a UAG codon, which is a stop codon. So then the polypeptide would stop at that point and terminate, and it would be a shorter polypeptide. This is one of the changes that occurs in the dystrophin gene to cause muscular dystrophy. It's a single change, changes a coding, a codon for one amino acid into a stop codon, and then truncates that protein so it lacks the function of attaching the actins and myosins to the membrane of that cell, so it no longer can contract as a muscle. One other type of mutation I should probably mention here is a change that changes an intron exon border. So this occurs occasionally in which the intron and exon borders are mutated. A cryptic exon intron border is used, and then the reading frame for that protein is disrupted. This occurs it is known to occur in beta thalassemias, which cause severe anemia, where the globin gene is not spliced correctly because of a mutation in that intron exon border. Okay, so that's all I have to say today about mutation. In a subsequent video, I'll talk about repair of some of these mutations, how they're repaired, and so it'll extend this discussion on mutation a bit. Okay, thanks. Talk to you a little later.